Well, let's talk about methicillin resistant Staphylococcus aureus. So first of all, what are the antibiotic susceptibility patterns of clinical Staphylococcus aureus isolates? So according to CDC, approximately 10% of Staphylococcus aureus isolates are susceptible to penicillin. However, many Staphylococcus aureus strains, while resistant to penicillin, they remain susceptible to penicillinase stable penicillins. And the examples of penicillinase stable penicillins to which these Staphylococcus aureus which are resistant to penicillin render susceptible are oxacillin and methicillin. Now, strains that are oxacillin and methicillin resistant is strictly termed methicillin resistant Staphylococcus aureus. They are also emerging and they are resistant to all beta-lactam agents including cephalosporins and carbapenems. Although they may be susceptible to the newest class of MRSA active cephalosporins, example ceftaroline. Now here in the comments you can see the cephalosporins are similar to penicillins in their mechanism of action, that is binding to penicillin binding protein and inhibiting cell wall synthesis, and they can be categorized from first generation to fifth generation. And here you can see the examples of different generations of cephalosporins. So here the methicillin resistant Staphylococcus aureus, they will be resistant to all of these cephalosporins except some of the fifth generation of cephalosporins, for example ceftaroline, and they will also be resistant to carbapenems, which is here in the comment. You can see here carbapenems are also beta-lactam drugs which bind to penicillin binding protein and inhibit cell wall synthesis. And few examples for carbapenems are imipenem, meropenem, and atrapenem. So the strains of MRSA causing healthcare associated infections are often resistant to other commonly used antimicrobial agents also, including erythromycin, clindamycin, and fluoroquinolones, while strains causing community acquired infections are often resistant to only beta-lactam agents, erythromycin, and occasionally to fluoroquinolones. What is frightening here is, since 1996, methicillin-resistant strains with decreased susceptibility to vancomycin, that is minimum inhibitory concentration of 4 to 8 microgram per ml, and strains that are fully resistant to vancomycin, which is determined by minimum inhibitory concentration, that is more than or equal to 16 microgram per ml, have been reported. This is quite frustrating because vancomycin was a choice of drug for methicillin resistant Staphylococcus aureus infections. So how should the clinical laboratories test for MRSA? In addition to growth microdilution testing, the Clinical and Laboratory Standards Institute, that is CLSI in short form, recommends the cefoxitin disc diffusion test, or a plate containing 6 microgram per ml of oxacillin in Muller-Hinton agar supplemented with 4% NaCl as alternative methods of testing for MRSA. So, cefoxitin disc diffusion method can be applied, or Muller-Hinton agar containing 6 microgram per ml of oxacillin that is also supplemented with 4% of NaCl can be used as some of the alternatives to broad type microdilution testing, and this is recommended, or yes, this is recommended by the CRSI. In addition, there are FDA approved assays for molecular detection of MAC-A gene and commercially available chromogenic agars that can be used for MRSA detection. Is it difficult to detect oxacillin or methicillin resistance? Well, MRSA is resistant to all beta-lactams because of presence of MAC-A, a gene that produces a penicillin binding protein, PBP2A, with lower affinity for beta-lactam antibiotics. So mechanism of oxacillin resistance other than MAC-A are rare. And so accurate detection of oxacillin or methicillin resistance can be difficult due to presence of two subpopulations. There is one susceptible to the beta-lactam antibiotics and other that is resistant to beta-lactam antibiotics. They may coexist within a culture of Staphylococcus. So all the cells in a culture may carry the genetic information for resistance, but only a small number may express the resistance in vitro. This phenomenon is termed as heteroresistance and occurs in staphylococci resistant to penicillin as stable penicillin such as oxacillin. So cells expressing heteroresistance grow more slowly than oxacillin susceptible population and may be missed at temperatures above 35 degrees Celsius. This is why CLSI recommends incubating isolates being tested against oxacillin at 33 to 35 degrees Celsius. The temperature should be 35 degrees Celsius at maximum for a full 24 hours before reading. So isolates tested against cefoxitin using either disk diffusion or broth microdilution should also be incubated at 33 to 35 degrees Celsius but can be read after 16 to 18 and 16 to 20 hours respectively. So are there additional tests to detect oxacillin or methicillin resistance? Well, yes, nucleic acid amplification tests such as polymerase chain reaction can be used for direct detection of MAC-A gene, the most common gene mediating oxacillin resistance in staphylococci. However, MAC-A PCR test will not detect novel resistance mechanisms such as MAC-C or uncommon phenotypes such as borderline resistance, oxacillin resistance. An alternative method for detection of MRSA is the use of anti-PVP2A monoclonal antibodies available as latex agglutination or immunochromatographic membrane assays. So, if we can detect penicillin binding protein 2A, which is an alternative for penicillin binding protein expressed by methicillin resistant staphylococci, if we can detect these altered proteins, yeah, by antigen antibody reactions using monoclonal antibodies, for example latex agglutination test or simple immunochromatographic membrane tests, then it is quite handy and it could be quite handy and very useful and very quick and also very less labor requiring method of detection of methicillin resistant staphylococci. Finally, there are commercially available chromogenic agars that we have already discussed. That can be used for MRSA detection. Here you can see the chromogenic agar is for isolation and differentiation of methicillin resistant Staphylococcus aureus. Methicillin resistant Staphylococcus aureus appear as rose to mauve colored colonies. So how is MAC-A gene involved in mechanism of resistance? As we have already discussed, staphylococcal resistant to oxacillin or methicillin occurs when an isolate produces an altered penicillin binding protein PVP2A, which is encoded by MAC-A gene, and the variant penicillin binding protein, which is PVP2A, binds to beta-lactam with lower avidity, which results in resistance to this class of antibiotic agent. So what are breakpoints for testing susceptibility of Staphylococcus to oxacillin? The CLSI clinical breakpoints for Staphylococcus aureus are different than those for coagulase negative staphylococci. For this, you can refer to CLSI approved standard M100 also. In short, oxacillin disc diffusion testing is not reliable for detecting oxacillin or methicillin resistant. So cefoxitin should be used as surrogate for disc diffusion testing for methicillin resistant staphylococcus, because oxacillin is considered as less stable. It is a stable drug in regards to storage, but lesser if compared to cefoxitin. And also cefoxitin is a antimicrobial agent that can induce the MAC-A gene in staphylococci in vitro, right? So cefoxitin is the best choice if disk diffusion test is considered. And so interpretive criteria in microgram per ml for oxacillin MIC test is, for MIC test, we're talking about the MIC test, so we are using oxacillin here. So for Staphylococcus aureus and Staphylococcus lugdunensis, it is if it is less than 2 microgram per ml, then it is considered susceptible, because only this proportion or this concentration, only less than 2 microgram per ml of oxacillin is killing the organism. The organism is appearing susceptible towards as low as 2 or less than 2 microgram per ml of oxacillin, so it is considered susceptible, and there is no value for intermediate here. And to call the staphylococci a resistant variant, it should be resistant to, equal to, or less than 4 microgram per ml. In other language, the MIC should be more than or equal to 4 microgram per ml. So for coagulase negative staphylococci, it is less than or equal to 0.25 microgram per ml, and no value for intermediate. And if it is 0.5 more than or equal to 0.5 microgram per ml, the coagulase negative staphylococci is considered resistant. So interpretive criteria in microgram per ml for cefoxitin MIC test. Remember, we're not talking about disk diffusion here. So Staphylococcus aureus and Staphylococcus lugdunensis, they are considered susceptible if the MIC is less than or equal to 4 microgram per ml, while there is no value for intermediate, and it is considered resistant if the MIC value is more than or equal to 8 microgram per ml, and there is no data for cons when cefoxitin MIC test is done. So interpretive criteria in MM for cefoxitin disk diffusion. Now we are talking about disk diffusion test, which is more common in laboratories. So cefoxitin disk diffusion test, the Staphylococcus aureus or Staphylococcus lugdunensis, they should produce a zone of inhibition more than or equal to 22 mm in order to call them susceptible, while zone of inhibition less than or equal to 21 mm, they are considered resistant. For coagulase negative staphylococci, them producing more than or equal to 25 mm of zone of inhibition are considered susceptible, while less than or equal to 25 mm are considered resistant. So why are oxacillin and cefoxitin tested instead of methicillin? It's a very interesting question, and the answer would be, first, methicillin is no longer commercially available in the United States. Secondly, oxacillin maintains its activity during storage better than methicillin and is more likely to detect heteroresistant strains. So oxacillin should have been used. Why cefoxitin? Because cefoxitin is an even better inducer of MAC-A gene, and tests using cefoxitin give more reproducible and accurate results than tests with oxacillin. That's why cefoxitin discs are used instead of methicillin, even though the organism are called methicillin resistant staphylococci. The name is only for the historical value, and this is all that I could grab from the website of CDC. I hope you enjoyed learning together with me. Thank you so much.